Hey guys, we're back with our next chapter in biology. So we're looking now at unit 1.3. Sorry, trying to get the fan out of the way. So we're looking at unit 1.3, the world of the cell. And we're going to start with part 1, which just kind of looks at a bit of an introduction to cells, cell theory, etc. So why don't you say, let's go get started, huh? There it is. All right, so first thing we're going to do is we're going to talk about 5 famous people who contributed to us discovering cells. So they're scientists, they've worked throughout the past few centuries, and it's thanks to them that we know what we do about cells. They laid all the foundation for it. So the first one we're going to talk about is Robert Hooke. He was an English scientist, and in the 1600s, he was working with some of the first light microscopes. Specifically, 1665, he was looking at thin slices of cork. Now, cork is a type of plant. It comes from a tree. And what he was looking at is he took very, 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 very thin cuts from it. And then he put those underneath the microscope, shone some light through it. And he was able to see what looked like little tiny rooms or chambers, kind of like in the picture here. Now, he saw thousands of these in his samples, and he named them cells. Now, the reason why he called them cells is because they reminded him of the little rooms in a monastery where the monks lived. And the monks called their little tiny rooms cells. So since they reminded him of those rooms, he called them cells as well. Then we have Anton Van Leeuwenhoek. And he was a Dutch scientist, and we're skipping forward to 1674. He was the first to actually observe microorganisms in pond water. So what he did was he took some of the samples from ponds, and he looked under them, and using the help of different microscopes and things of that nature at the time, he was able to see these little tiny things swimming around in it. And they were things you can't see with your naked eye, but it was him observing them with some of the newer technology of the day. He was able to see them. And as you can kind of see here at the bottom, they show you a little bit about what we're talking about. So his diagrams would be this one right here. And then these kind of go along with it. Now, he didn't know exactly what they were. He called them animalcules. So kind of like if you take the word animal and molecules and put them together, we have a lot more in-depth knowledge about these organisms today, these microscopic organisms. All right, next we are going on to Matthias Schleiden. He was a German botanist, and a botanist is a scientist who studies plants. Now around 1838, he was looking at samples of plants underneath microscopes. And what he found from every single sample he looked at is that every sample showed it was made out of cells, whether it's the leaves, the stems, the roots, the flower, the fruit. And he had samples from all over. And he was looking at all these different samples, and he noticed a commonality that they all contained cells. They were all made out of cells. So based on these observations, he was the one who concluded that every plant had to be made up of cells. Every single plant across the whole planet had to be made out of cells. Theodor Schwann was a German physiologist. So a physiologist is someone who works with like tissues and anatomy, things like that, how body systems work. And in 1839, he concluded that all animals had to be made of cells as well. So he worked in a very similar fashion as Schleiden, but instead of looking at a whole bunch of different plant samples, he looked at a whole bunch of different animal samples. So samples from different animals, different types of tissues from the same animal. And what he noticed is that, again, all cells. So whether it was the skin of a rabbit or the lung of a lizard or the heart of a fish, all of them were made out of cells. So he concluded that all animals had to be made out of cells as well. And our last one, we have Rudolf Virchow. So Rudolf Virchow was a German physician, so a doctor, and he studied cell reproduction. Now, for a while, people couldn't exactly figure out where cells came from. There was the idea of spontaneous generation or regeneration, sorry. And what that referred to is people noticed that if, meat was left out too long, then boom, maggots just suddenly emerged from it. So it was thought that there might have been something in the air that could lead to the development of cells. Well, through a series of experiments involving putting meat in open containers, putting meat in containers with mesh on it or some kind of semi-permeable covering, and putting meat in a completely covered container, what they found is that flies always came to the meat first. So the meat that was left open, a lot of flies flew around it, they came down, they fed on the meat, and then they left. The one with the mesh, only some of the flies could get in, and then they left. No flies could get to the one with the sealed cover. And then not long after that, that's when the maggots would develop and show and come out of the meat. And the meat that was in the container that was completely open had a lot of maggots, which now we know makes sense because there was a lot of flies that visited it. The one with the mesh lid had only a little bit of maggots come out, and the one that was completely sealed off had no maggots. So what they instead realized is that the maggots weren't just spontaneously coming together out of nothing and forming into these maggots and coming from nothing, but it was the eggs of the flies. The flies, when they're feeding, left eggs there, and then those eggs hatched into the maggots. So what that had to tell us then, and what Virchow figured out, is that if cells aren't just coming out of nothing, they're not just spontaneously coming out of nothing, then what must they come from? So he observed cells, and what he determined was that cells go through their own form of reproduction, and that all cells must come from pre-existing cells. So one cell would become two. Two would divide to become 4, 4 to 8, 8 to 16, and so on and so on. So cells don't just poof appear, they actually come from existing cells. Just like how humans didn't just poof appear, we came from existing humans, our parents. It's the same idea. All right, so let's talk a little bit about the cell theory. Now, the cell theory still holds today. There are 3 main points to the cell theory, and we use this as a guiding light because it applies to all cells. So cells are the basic unit of structure and function in living things, meaning they're the smallest thing that we consider to be alive, and also they're the smallest functional unit. A cell by itself can live, do all of its different functions. We know that because there's plenty of single-celled organisms. They can also come together and function together in order to create a larger multicellular organism. Right? All living things are composed of cells. So if it's alive, it is made of cells, whether it is a dog, a fish, a human, a tree, bacteria. If it's alive, it has to be made out of cells. And number 3, new cells are produced from existing cells. So that's Virchow's contribution. All right. Let's talk a little bit about the basic structure. So at this point, we're just going to name 3 things. Our entire next section of notes will be on all the different organelles and their function. So 3 of the most basic structures within cells. First off is the cell membrane. It's the outer layer that surrounds and protects the cell. It is a thin layer, it's very, very flexible, and it's semi-permeable, meaning only some things are allowed to cross it to go in and out of the cell. Then there is cytoplasm, a gel-like material inside the cell. This holds and supports the different organelles within the cell and keeps them, you know, afloat and able to move around and do their jobs. Now, all cells have a cell membrane and cytoplasm. Not all cells have the next one, a nucleus. There are a certain classification of cells that do not have a nucleus. We'll get to that in a moment. But what a nucleus is, it is an organelle that contains the genetic information, so it holds the actual DNA of that cell. And because it holds the DNA, we say that it controls the cell activities. Now, the only reason why we say it controls the cell activities is because the DNA is the blueprint on how to run the cell, how to do all the different things, make all the parts of the cell, do all the jobs of the cell. And as we said, some cells have one and some cells do not. All right, so cell classification, there's two main groups of cells, prokaryotic cells and eukaryotic cells. Now the picture up here, let me just move my camera a little bit so it's out of the way. There we go. So this picture up here is an example of a prokaryotic cell. It has no nucleus and no membrane-bound organelle. So it's a smaller cell. It's very, very simple. There's not a whole lot going on. So you can see on the inside, not much going on. Since there's no nucleus, that means that the DNA is not contained in anything. It's just sitting right there in the cytoplasm. And since it's sitting in the cytoplasm, and this is a smaller, simpler cell, it doesn't have to make all these other organelles or make proteins to help the organelles function. We actually notice that their DNA is shorter. It tends to be smaller and more simpler than eukaryotic organisms. Give me a moment, Ripley. Come here, dude. Yeah. Not only that, but instead of being linear, the ends of prokaryotic DNA actually bond together, they fuse, and it forms circular DNA. Let me finish with this. Come here. Sorry. So to go along with that, all prokaryotic cells or prokaryotic organisms are single-celled. And the best example, any kind of bacteria. Here, come here. Yeah, little fluffy brat. Give me a moment. Yes, I call him a fluffy brat. So all bacteria are prokaryotic organisms. Now, let's take a look at eukaryotic organisms and eukaryotic cells. That's this picture at the bottom. Notice how there's a lot more going on. There's a lot more structures in there. So there's a nucleus that holds the DNA. The DNA is longer and it's linear. The ends do not fuse into a circle. They also have a lot of other membrane-bound organelles, such as mitochondria and the Golgi apparatus, the smooth endoplasmic reticulum, the rough endoplasmic reticulum. Those are present in here. We have a whole lot of different organelles, and they all have very, very particular functions, very specific jobs. Eukaryotic organisms can be single-celled or multicellular. There are some single-celled organisms that are eukaryotic. There's also a lot of multicellular ones like us. We are eukaryotic organisms. Our cells are eukaryotic cells. We said that the bacteria were the prokaryotic organisms. Well, eukaryotes includes animals, plants, protists, and fungi. So we have a wide variety of different things. Again, going all the way from single-celled to multicellular. So we have much larger, much more complex cells going on in eukaryotic organisms. Yeah, that's right, because I moved this. Let's put that back. All right, now let's talk about our additional resources. So we had a Science in Pajamas video by me where we kind of compare and contrast prokaryotic versus eukaryotic cells. So if you get the chance, go ahead and check that out. We also have some other videos from TED Ed. We have the wacky history of the cell theory. So it talks about some of the people as well as the cell theory itself. Khan Academy talking about the different aspects of the cell theory along with again, scientists involved in it. And then we have the Amoeba Sisters. I love these ladies. Anyways, it's them talking about the differences between prokaryotic versus eukaryotic cells. All right. Well, that pretty much sums it up. You guys have any questions? Catch me in our Google Classroom, email, or actual class, depending on when you're viewing this. But yeah, if you have questions, just let me know, you guys. In the meantime, stay awesome, stay curious, and most importantly, stay safe and healthy. All right, you guys, take care. Bye-bye.